Hello, science enthusiasts and curious minds. Today, we venture into the captivating world of genetic engineering, where the threads of life's intricate tapestry are rewoven with the threads of innovation. In this thought-provoking video, we delve deep into the realms of biotechnology and genetic manipulation, uncovering the remarkable power humans have harnessed to reshape the very fabric of life. Prepare to embark on a journey that explores both the marvels of scientific discovery and the ethical considerations that accompany our ability to design life itself. Genetic engineering is a scientific field that uses genetic modification to produce desired traits in living organisms. Examples include the creation of vaccines, human insulin, and other medications through recombinant DNA technology. The term genetic engineering also refers to direct changes made to the DNA of an organism, including selective breeding, creation of transgenic organisms, those with genes from different species, and mutation breeding, where random mutations are induced in plants or animals. Genetic engineering is the process of altering the DNA of living organisms. It's been used to create plants that produce drugs, animals that produce human proteins in their milk, and bacteria that can digest toxins. Genetic engineering has many ethical implications, and it could have benefits and risks for humans as well as other organisms. This article will discuss some of these implications, along with potential applications for genetic engineering in medicine and agriculture. Transgenesis is an experimental technique in which new genes are introduced into the genome of an organism. The new genetic material can be transferred from one organism to another, or it can be inserted randomly into the genome of a single-celled organism like bacteria or yeast. In transgenic animals, researchers use viruses and other vectors to deliver the foreign DNA that they want to insert into their target cell's genome. Vectors are small pieces of DNA that carry other genes into cells. They're used because they're easy to manipulate and produce large quantities of DNA quickly, two qualities essential for making transgenic animals at scale. The most common type of vector used in transgenesis is called an episome. It's made up of circular pieces of DNA that don't integrate themselves permanently into a cell's genome, but instead sit on top like hats on heads. Episomes can stay there indefinitely without affecting their host's health. Once scientists have finished editing them with their new genes, however, either through random insertion or targeted insertion using CRISPR-Cas9 technology, they'll need some way for these changes not only stay put, but also get passed down from generation after generation without disappearing over time, thanks largely due outbreeding among related individuals during reproduction. Cloning is the process of duplicating an organism. The DNA of an organism is copied, and this copy is inserted into an egg cell, which is then implanted into a surrogate mother. The baby that results from this process will be genetically identical to its parent. The first animal cloned from adult cells was Dolly, a sheep born in 1996. She lived for 6 years before dying from lung disease caused by pneumonia contracted after she was exposed to anesthesia during medical testing at age 4. She had been diagnosed with arthritis. Gene splicing, gene editing. Gene splicing is the process of combining different DNA sequences to create a new gene. Gene editing is the process of making changes to an existing gene. Both techniques are used in genetic engineering, and both can be used on their own or together to produce desired results, but they're not exactly alike. Gene splicing involves cutting up pieces of DNA and then pasting them back together in new combinations. It's like putting together a puzzle with pre-cut pieces instead of having to cut out all your own pieces from scratch. On the other hand, gene editing involves making specific cuts at certain points within an organism's genome, its complete set of genes, and then allowing nature to take its course after that point has been altered. This method allows researchers more control over what happens next, but also requires more extensive knowledge about how an organism works than does simply inserting foreign DNA into its genome via recombinant methods such as transfection or viral infection. Gene knockout, knock-in, knock-out. Gene knockouts are used to create mutations that can be studied. The gene is removed from its normal location and replaced with a piece of DNA that has no function, so it doesn't produce a protein. This causes the organism to have an abnormal phenotype. The goal is to observe how this mutation affects the organism's ability to survive or reproduce. Gene knockouts can also be used therapeutically. If you know what protein does what in your body, you might be able to target drugs at specific proteins that cause disease symptoms. Bioengineering and synthetic biology. Synthetic biology is a new field that combines engineering and biology. It uses engineered biological systems to solve problems, develop new technological applications, and produce useful products. The term synthetic refers to the fact that scientists create these engineered systems in their laboratories. They don't find them in nature. Bioengineering and synthetic biology use similar methods but have different goals. Bioengineering aims to improve existing organisms or create new ones with specific traits. Synthetic biology seeks to understand how living things work at a molecular level so we can build them from scratch using information encoded in DNA or RNA molecule. Genetic engineering is an area of intensive research with many implications. Genetic engineering is an area of intensive research with many implications. It can be used to solve problems such as those related to disease, but it also raises ethical concerns. Genetic engineering is a powerful tool that can be used to create new products and services. Genetic engineering is an area of intensive research with many implications. It has the potential to change everything from how we treat diseases to how crops are grown. The rise of synthetic biology and bioengineering also means that we may soon be able to create new life forms from scratch by combining DNA sequences from different organisms or even making entirely new genes ourselves. As we bring our exploration of genetic engineering to a close, we find ourselves at the crossroads of remarkable discovery and profound responsibility. If the intricate dance of genes and the boundless potential of genetic engineering have ignited your curiosity, consider showing your appreciation by giving that likes button a well-deserved click. And for those who are as intrigued as we are by the mysteries of life's code and the possibilities it holds, don't forget to subscribe to our channel for more enlightening journeys into the frontiers of science and technology. Thank you for joining us on this enlightening quest. And remember, the code of life is in our hands. Until our next exploration, keep exploration, keep exploring, and keep shaping the future with the power of knowledge. Safe travels on this extraordinary genetic journey.